Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor and Head of the Department of Biochemistry, St. Joseph's College for Women. Through this video, we'll be studying HMP shunt and regulation. The topics that will be covered include basic introduction, followed by location of the HMP shunt pathway where it takes place, then the reactions of the pathway and the various phases of it, then the importance of pentoses, importance of NADPH, and then regulation of HMP shunt pathway. Hexose monophosphate pathway or HMP shunt. It is also called as pentose phosphate pathway or phosphogluconate pathway. We see that this is an alternative pathway to glycolysis as well as the TCA cycle for the complete oxidation of glucose. And HMP shunt is more anabolic in nature because it is mainly concerned with the biosynthesis of NADPH and pentoses. It is also called as a multifunctional pathway because it starts with glucose 6-phosphate and here there is no ATP usage or production in this pathway. So it is multifunctional because there are several interconvertible substances which are produced in this pathway in different directions in the metabolic reactions that you observe here. Coming to the location of the pathway, the enzymes of HMP shunt, they are located in the cytosol region. Many tissues are involved in this, like the tissues such as liver, adipose tissue, adrenal gland, of the testes erythrocytes, then even the lactating mammary glands. All these, they are very much active in this HMP shunt. And most of these tissues, they are involved in the biosynthesis of either fatty acids or in the biosynthesis of steroids, which are dependent upon the supply of NADPH. If NADPH is there, their synthesis will be taking place. So, in all these tissues, you find HMP shunt happening. The reactions of the HMP shunt pathway, they are divided into two phases. Like you have two phases of reactions. One is the oxidative phase and then the non-oxidative phase. Coming to the oxidative phase. The oxidative phase of HMP shunt, it is an irreversible metabolic process and it mainly occurs in the cytosol. So how does it begin? It begins with the oxidation of glucose 6-phosphate. Here glucose 6-phosphate, it is oxidized into 6-phosphogluconolactone by the enzyme glucose 6-phosphate dehydrogenase. This step, it is the rate limiting and regulatory point of the pathway, and the resulting one which is formed is 6-phosphogluconolactone. Along with that, you get NADPH, which is formed as a result of reduction of NADP+. The activity of this glucose 6-phosphate dehydrogenase, it is strongly influenced by the presence of NADP+ as well as NADPH. So high levels of NADP+ will stimulate this enzyme, while high levels of NADPH will act as feedback inhibitors. The lactone intermediate which is formed, it is hydrolyzed by the enzyme lactonase to form 6-phosphogluconate, which is a linear carboxylic acid. This compound, it undergoes further decarboxylation catalyzed by the enzyme 6-phosphogluconate dehydrogenase, and it results in the formation of ribulose 5-phosphate, and you see that another molecule of NADPH is produced. along with release of carbon dioxide. So overall, for every molecule of glucose 6-phosphate that enters this oxidative phase, you see that there are two molecules of NADPH which are produced. Along with that, one molecule of ribulose 5-phosphate is generated. NADPH, it is the vital reducing agent that is used for various anabolic process and in maintaining even the redox state of the cell, especially the red blood cells, and it also helps prevent oxidative damage of red blood cells. The second phase is the non-oxidative phase. The non-oxidative phase of HMP shunt, it is reversible and it plays a crucial role in the interconversion of sugar phosphates, allowing the cells to adapt to its metabolic needs. It begins with the conversion of ribulose 5-phosphate, which is formed in the oxidative phase, into either ribose 5-phosphate or xylulose 5-phosphate. So the formation of ribose 5-phosphate from ribulose 5-phosphate is because of the enzyme ribulose 5-phosphate isomerase, while the synthesis of xylulose 5-phosphate from ribulose 5-phosphate is because of the enzyme ribulose 5 epimerase. So you have an isomerase and an epimerase that will decide which is formed. And next here you see that the ribose 5-phosphate, it is very important and it is used in nucleotide synthesis as well as nucleic acid synthesis. And phosphopentaisomerase, it forms this xylulose 5-phosphate. Now the pathway further proceeds with a series of carbon shuffling reactions involving enzymes like transketolase, transaldolase, which transfer 2 carbons and 3 carbon units between sugar molecules. In the first major transketolase reaction, the xylulose 5-phosphate and ribose 5-phosphate, they are converted into glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. Here you have transketolase which is doing the process. Next, we have transaldolase which acts on this. The transaldolase enzyme acts on these products and it will result in the formation of fructose 6-phosphate and erythrose 4-phosphate. A second ketolase reaction follows this where another xylulose 5-phosphate reacts with the erythrose 4-phosphate to produce additional molecule of glyceraldehyde 3-phosphate and fructose 6-phosphate. So you have here one fructose 6-phosphate which is formed while erythrose 4-phosphate combines with xylulose 5-phosphate to produce glyceraldehyde 3-phosphate and fructose 6-phosphate. So these are the final products. So what are the final products? Fructose 6-phosphate and glyceraldehyde 3-phosphate. The fructose 6-phosphate and glyceraldehyde 3-phosphate, they can enter glycolysis and can produce energy or can be redirected into gluconeogenesis depending upon the cell requirements. This phase is very important when there is a need for NADPH. When the cell needs high amount of NADPH, this will be taking place. But the requirement for ribose 5-phosphate as such is low, allowing cell to recycle carbon skeleton efficiently. So let us see the summary. Glucose 6-phosphate is converted to 6-phosphogluconolactone by the enzyme glucose-6-phosphate dehydrogenase, resulting in the formation of NADPH. Now this phosphogluconolactone is converted to 6-phosphogluconate by the enzyme 6-phosphogluconolactonase. Now the 6-phosphogluconolactone is converted to ribulose 5-phosphate by 6-phosphogluconate dehydrogenase, where you have one more NADPH formed and one molecule of carbon dioxide is released. Now from here you have the formation of either ribose 5-phosphate or ribulose 5 or xylulose 5-phosphate. So ribose 5-phosphate is formed by the enzyme ribulose 5-phosphate isomerase, while xylulose 5-phosphate is formed from ribulose 5-phosphate isomerase. So these two can lead to the formation of, in the presence of transcrystallase enzyme, can lead to the formation of glyceraldehyde 3-phosphate and C2-heptulose 7-phosphate. So here C2-heptulose 7-phosphate is further acted upon by transaldolase, resulting in the formation of fructose 6-phosphate and erythrose 4-phosphate. So here you have fructose 6-phosphate which enters the glycolysis. You also have this fructose 6-phosphate and erythrose 6-phosphate. They can also give rise to fructose 6-phosphate and glyceraldehyde 3-phosphate. So we know that whatever fructose 6-phosphate that is produced, it enters the glycolysis, while the erythrose 4-phosphate combines with xylulose 5-phosphate to form fructose 6-phosphate and glyceraldehyde 3-phosphate. So this is how this pathway operates. Finally, the end products will be fructose 6-phosphate and glyceraldehyde 3-phosphate. So when you take glucose 6-phosphate in the presence of 12 NADP+, it is converted to 6 molecules of carbon dioxide. That means each carbon gets eliminated and finally you get 12 NADPH molecules. So this pathway, it is considered to be very important for the synthesis of pentoses. Pentoses, particularly ribose 5-phosphate, it is a very important 5-carbon sugar which is produced in the non-oxidative phase of HMP shunt. These sugars, they play very important role in nucleotide and nucleic acid synthesis. So ribose 5-phosphate, we know that it is the precursor for purines and pyrimidines. That means it's also important for the synthesis of DNA, RNA, ATP, NAD+, FUD, coenzymes, etc. The HMP shunt, it ensures that dividing cells such as those of bone marrow and even skin, the gastrointestinal tract, all these have a steady supply of this ribose 5-phosphate to support the rapid DNA synthesis and even RNA synthesis. Additionally, the reversible nature of non-oxidative phase, it allows cell to produce ribulose 5-phosphate even from the glycolytic intermediates like fructose 6-phosphate and even glyceraldehyde 3-phosphate when glucose 6-phosphate is scarce and or even when NADPH is not required in large amounts. Now coming to the importance of NADPH, the nicotinamide adenine dinucleotide phosphate, which is a reduced form of NADP+, this acts as a very important reducing agent in the cellular metabolism and is produced during the oxidative phase of HMP shunt, the first phase of HMP shunt. So its main functions are reduced biosynthesis, maintenance of reduced glutathione, detoxification reactions, as well as immune defense. First one is reductive biosynthesis. NADPH, it has a very important role in many anabolic reactions. It provides reducing power that is required for anabolic reactions like the fatty acid synthesis, which takes place in the liver, in tissues like liver, adipose tissue, so on and so forth. And it is also required for cholesterol synthesis in liver and adrenal cortex, and even steroid hormone production. It is facilitated by NADPH in the adrenal cortex, and even in the mammary glands. It plays vital role, mainly lactating mammary glands. NADPH, it is required for maintaining the glutathione in reduced state. So in red blood cells and other cells, when they are exposed to oxidative stress, then NADPH, it helps maintain the glutathione in reduced form. Reduced glutathione, it protects the cells by neutralizing the reactive oxygen species, ROS, like hydrogen peroxide, and preventing them from damaging cellular proteins, lipids, as well as DNA. Third importance is in the detoxification process in the liver. So NADPH, it acts as a cofactor for cytochrome P450. So we know that cytochrome P450 monooxygenase systems, they are seen in the liver, and they are involved in the detoxification of many drugs as well as xenobiotics. They'll be detoxifying them. In immune defense, NADPH oxidase cycle, it helps here. So you have this phagocytic cells such as the neutrophils and macrophages. The NADPH is used by the enzyme NADPH oxidase. In this one, it generates superoxide free radicals during the respiratory burst. These radicals, they'll be helping to kill ingested pathogens. So this NADPH, it becomes very much indispensable, not just for the anabolic process, but also for cellular defense mechanisms and even the redox balance. Coming to regulation of HMP shunt pathway. So this regulation is by 5 methods. You have regulation at rate limiting step, feedback regulation by NADPH or NADP plus ratio, hormonal regulation, tissue specific activity, regulation based on cellular demands. First regulation is regulation at rate limiting step. So the primary regulation of HMP shunt pathway, it occurs at the rate limiting enzyme, which is glucose 6-phosphate dehydrogenase. This enzyme, it catalyzes the first step of oxidative phase wherein the glucose 6-phosphate is converted into 6-phosphogluconolactone in the HMP shunt, and it also simultaneously results in the production of NADPH. It is produced as the byproduct. Since this step commits glucose 6-phosphate to the HMP shunt, its regulation determines the overall activity of the pathway. The activity of glucose 6-phosphate dehydrogenase is tightly controlled by the availability of NADP plus and then NADPH, ensuring that the pathway, it activates only when the reducing equivalents are required. That means only when NADPH is required. If NADP plus is in scarce, this pathway will not operate. If NADP plus is in surplus, then only it will operate. Or if NADPH is in excess, it will not operate. Feedback regulation by NADPH or NADP plus. So here it is the key enzyme, that is glucose 6-phosphate dehydrogenase, which is allosterically regulated by the levels of NADP plus and NADPH. When NADP plus levels are high, it indicates that the cell definitely needs these reducing parts. Thus, it activates the enzyme and simultaneously the oxidative phase of HMP shunt is initiated. In contrast, if NADPH levels are increasing, if they are high, it exerts a negative feedback on the particular enzyme, on this enzyme, and thereby it slows down the pathway, and then it ensures that NADPH synthesis and its accumulation will not be taking place. So if excessive is there, it is accumulating, and here thereby NADPH, it ensures that it is synthesized according to the need. That means particularly during reductive biosynthetic reactions if they are taking place, and during oxidative defense mechanisms. The third is hormonal regulation. Insulin role in HMP regulation. Insulin, it plays very important role, the hormone role in regulating HMP pathway. In tissues such as liver and adipose tissue where fatty acids, cholesterol biosynthesis occurs, insulin regulates the expression of this glucose 6-phosphate dehydrogenase enzyme and even the other enzymes of HMP shunt. After carbohydrate rich meal, what you see is that the insulin levels are increased and the glucose levels are also increased. So thereby the glucose enters into the cell, increasing the glucose 6-phosphate levels in the cell. So here at this point, insulin upregulates the gene expression of glucose 6-phosphate dehydrogenase. So here the HMP shunt pathway is activated and NADPH is produced, and NADPH is also required for lipid biosynthesis also. So lipids are synthesized if glucose is high. Lipids are also synthesized in the liver and adipose tissues. Tissue specific activity. The activity of HMP shunt, it is not the same in all the tissues. It varies from tissue to tissue based upon the demand for NAD+. So it is like in the liver, it is supporting biosynthesis and detoxification. In the adipose tissue, it is contributing to fatty acid synthesis. In adrenal cortex, it is facilitating steroid hormone synthesis. In RBC, it is used for maintaining the glutathione to combat the oxidative stress and combat free radicals produced. In other tissues, other tissues exhibit low levels of this HMP pathway. So in all these tissues, based upon the demand for NADPH and ribose 5-phosphate, it will be operating. Regulation of pentose phosphate pathway based upon the cellular demands. This pathway is also regulated according to the cell's specific metabolic needs. When the cell primary requirement is NADPH, say if the cell has identified that it requires it, it is needed for oxidative phase, then it is activated. The full oxidative phase is activated, and the pentose sugars formed, they are recycled. If these are formed, they are recycled again into the glycolytic intermediates. The pentose sugars, they are recycled there. Then ribose 5-phosphate demand, if it is there, ribose 5-phosphate, it is needed when the non-oxidative phase, if this is there, non-oxidative phase, it runs in reverse way. And even you have ribose 5-phosphate generation which is taking place, it's also generated from the glycolytic intermediates. So you see that ribose 5-phosphate, it is generated during cell division or nucleotide synthesis. And here, when it is required, the non-oxidative phase, it can run in the reverse way, and glycolytic intermediates like fructose 6-phosphate, glyceraldehyde 3-phosphate, they can also be used. So whatever products that we have seen in the non-oxidative phase, that is fructose 6-phosphate and glyceraldehyde 3-phosphate, since that phase is reversible, that can be used in the reverse way for the synthesis of ribose 5-phosphate without even engaging the oxidative phase. So this is how we can get ribose 5-phosphate when demand is there, running it in a reverse way, okay, without touching the oxidative phase. The textbooks that are used while preparing this material include The Principles of Biochemistry by Leninger, then Biochemistry by U.S. Narayana, then even Harper's Biochemistry. So what we have seen in this video is basic introduction to what is HMP pathway, its importance, where it takes place, the phases we have seen, the reactions and all. And then we have seen, studied the importance of mainly the pentoses as well as NADPH we have seen. And then the regulation of HMP shunt pathway. Thank you.